they are present in the basal surface of the seminiferous tubules. So what happens is the Satori cells, as they're forming the barrier, they are dividing the region into a basal part and a luminal part. So in the basal part, we have these spermatogonia. And then the luminal part, we have the actively dividing cells. So there are two types. We have dark cells and pale cells. The pale spermatogonia, they are known as type B cells, and they are the ones which are further participating in the whole procedure. Whereas the dark ones, type A, they are just simply undergoing mitosis and replacing and replenishing the stores. So what happens is that this type B pale spermatogonium, this has 46 chromosomes, diploid cells, undergoes maturation to form a primary spermatocyte. This is a simple mitotic division. This is known as spermatocytogenesis. Cytogenesis is the process when spermatogonium gets converted into a primary spermatocyte and nothing happens. There is no reduction in chromosomes. It's a simple mitotic division. But after that, this primary spermatocyte, which is still 46 chromosome diploid cell, then undergoes reductional division, which is meiosis 1, and it forms two secondary spermatocytes, which are now haploid. So which two cells are diploid? Spermatogonia and primary spermatocytes. Just like we had in oogenesis, we had oogonium and primary oocyte. Both of those were diploid, and now haploid cells are the secondary spermatocytes and the further spermatids. Now what happens is that another division occurs, which is meiosis 2, when we have the formation of 4 spermatids. Now what happens is that this whole procedure of spermatogenesis takes 74 days, which is also another important question. And what happens is that these spermatids, they are later converted into sperms, and this maturation is now known as spermiogenesis, and this takes separate 14 days. So we had spermatocytogenesis, which is a simple maturation, which occurred as a first step, and now at the end we have spermiogenesis. Now there's another term which is known as spermatidogenesis. This is the conversion of spermatocytes, which means this whole process from primary spermatocyte, this whole process from spermatocytogenesis onwards till the formation of spermatids. This is known as spermatidogenesis. So we have a total of 4 terminologies. Now what happens is that as the sperm are formed, now as they mature sperm, so what happens during maturation? The nucleus undergoes condensation, acrosomal cap is formed, tail is formed, and mobility is attained. So initially, the spermatid or the immature sperm was like this. It had a nucleus, it had mitochondria, it had Golgi complex, it had a membrane, and it had 2 centrioles. This is a very important question, that out of these 2 centrioles, one goes into the neck in the posterior there's a posterior curvature along the neck where one centriole is present, and the other centriole goes into the tail, and it has axial filaments. So this is a very important question, but there are the 2 centrioles in the mature form. And now these mitochondria form the middle piece, and now this Golgi complex is from the acrosomal cap. So what happens is, this is the mature one. So this is the end product of the whole spermatocytogenesis, the spermiogenesis, 74 plus 14 days. So the size is 40 to 60 microns, and lifespan is of 48 to 72 hours. Now head has the acrosomal cap, which is going to release the hyaluronidase enzyme, which is going to dissolve the corona radiata cells. With the neck, which is going to have one of the centrioles in its posterior aspect, and now we have the middle piece, and then we have the tail. Tail has and the other centriole, which is responsible for mobility. Now, very important thing is that in the seminiferous tubules, they are still, even after spermiogenesis, they are still immature. So from the tubules, from the seminiferous tubules, they go into the ready stage. From there, they go to epididymis. Now epididymis has a head and a tail. In the head, they become mature, because in the head we have brain, which makes us mature. So in the head, they attain their maturation, and in the tail, they attain their motility. So what helps to attain motility? Beta defensin protein. So if it's deficient, then there will be motility issues, and this maturation separately takes further 14 days. So we have a 74-day span, then we have a 14-day span, then we have another 2 to 14-day span. Now after being emptied into the cervix, they undergo capacitance. This is another procedure that happens, and this procedure, so they undergo capacitation, and what happens during capacitation is that they become hypermobile, and their glycoprotein coat gets removed, and their seminal plasma proteins are released. So these 2 things happen during capacitance.